Hello everyone and welcome to part 2 of your digestive system lecture. So we're going to pick back up on slide, I believe this was 37. Yep, 37, where we will discuss the celiac phase of digestion. So when you hear the word celiac, the first thing that should come to mind is the head. So when we discuss the head, it's more of the nervous system control of digestion. So when we first even start thinking of food, so think of your favorite food. Maybe it's something really sweet. Maybe you are a dessert person. Maybe it's more about salty snacks like French fries, chips, pizza, hot dog, tacos, whatever it is. Chances are, as I'm describing these different food types to you, your salivary glands may start secreting saliva within your mouth. I know it is for me right now. And so what we can attribute that to is this celiac phase. So the even the sight or the memory or thought of food is going to start triggering your parasympathetic nervous system and getting the body ready to digest the food that you're about to ingest. So the taste or smell of food and the tactile sensations of food in our mouth are all stimulating our medulla oblongata. And remember back to our nervous system lecture, this is found in the brain stem. So from the medulla oblongata, we are going to have a cranial nerve called the vagus nerve. This is cranial nerve number 10 that will extend off and has some parasympathetic nervous system fibers coursing through it. And so the vagus nerve is actually the longest of all the cranial nerves because it is traveling from the brainstem down through the region of the neck, down through the thorax and into the abdomen to give supply to abdominal organs. So it supplies our stomach here and activates our enteric plexus. Remember, our enteric plexus is made up of that submucosal plexus as well as the myenteric plexus that are found in the submucosal layer and muscularis externa layer respectively. So what's happening once it stimulates this area? The neurons within the stomach here are going to cause those chief and parietal cells that we found in the mucosal lining of the stomach to release hydrochloric acid and pepsin. And it'll also cause the secretion of gastrin and histamine. So what gastrin does is it goes into circulation and comes back to the stomach to cause more secretions. And also we will have the histamine stimulate more hydrochloric acid and pepsin. So now we're going to move into our gastric phase. And so the food at this point within our stomach has caused distension of the stomach, and that causes a parasympathetic reflex. So the action potentials of our vagus nerve gives us sensory input to our medulla oblongata. And then the medulla oblongata will send those parasympathetic nerve fibers back down to the stomach in order to tell it to cause more secretions within the stomach. It's also going to have some local reflexes take place due to this distension here. And so this in turn will also cause a release of the gastrin within the stomach, and it does the same thing here, goes through the circulation and back to the stomach, and it also releases histamine once again, and that will cause more secretion of the hydrochloric acid and the pepsin. So you could see some overlap between the cephalic phase and the gastric phase, but the major difference is what is causing the end result. With the cephalic, it was more of a input of the taste, the smell, the thought of foods. So again, more of that cephalic, that thought that's taking place in your mind, or you have the food within your mouth. And then the gastric phase, it's more of the distension within the stomach that is triggering the end result that we described. So the last phase here is going to be our intestinal phase. So at this point, we are going to move the chyme, that liquefied food, into the first part of the duodenum. And we have a pH of less than 2 or containing lipids that's going to start to inhibit the gastric secretions through 3 mechanisms. So those 3 mechanisms are all going to work to increase, they're all going to work to decrease the gastric secretion. So let's look at our first mechanism, and that is going to be the sensory input. It's always going to be due to these decreased levels of pH, so more of an acidic pH, or lipids being present within the chyme in the duodenum. So that sends that sensory input into the medulla from our duodenum and is going to inhibit this motor output being sent through our vagus nerve, telling it to increase secretions. So it inhibits that, and we are going to stop secretion of our pepsin and hydrochloric acid. The second mechanism is the local reflexes will inhibit the gastric secretion within the stomach. And the third mechanism is we get a release in the duodenum of hormones secretin and cholecystokinin that move through our blood circulation and over to the stomach to tell it to decrease its gastric secretions as well. So here's a nice table that gives a summary to those hormones we just discussed. We have our gastrin that's secreted from the stomach, and that is going to be stimulated through distension of the stomach due to the food being in there. Also, it has stimulation from digested proteins, stimulation from our autonomic nervous system, or if we ingest any alcohol or caffeine. Its job is going to be to increase gastric secretion, and then we have this category of motility effect. So motility is referring to the muscles that surround the stomach. How much is the stomach churning? And gastrin is going to cause a minor increase in the gastric motility. Our next hormone is secretin that was released by the duodenum, like we just said in the previous slide, and it's stimulated by the acidity of that chyme, so the pH of less than 2, and that'll cause a decrease within the gastric secretion. It'll also cause stimulation of pancreatic and bile secretions that are high in bicarbonate ions. So we'll talk more about this when we discuss these accessory organs of the digestive system, but basically we want that pH to move from less than 2 to more of the alkaline or basic side of the pH scale. And then this is going to just cause a decrease within gastric motility. Next we have the hormone cholecystokinin. This is also released by the duodenum, and its stimulation is the fatty acids and peptides that it finds within the duodenum, and that'll cause just a slight decreases within the gastric secretion. And then once again, we'll talk about some of this more in detail in our accessory organ section, but it's going to stimulate pancreatic secretions that are high in digestive enzymes and also cause a contraction of the gallbladder and relaxation of the hepatopancreatic sphincter. I know you don't know what that is yet, but just put a pin in it and come back to it after you listen to the portion on our accessory organs. And then as far as motility, it's going to strongly decrease the gastric motility. So now let's discuss the movements of the stomach. This is going to be a combination of mixing waves, which will happen 80% of the time, and peristaltic waves that'll happen 20% of the time. And at this point, both the esophageal sphincter pictured here and the pyloric sphincter pictured here are going to be closed and contracted. So we see that the mixing wave is initiated in the body of the stomach. Remember, that's this portion in here. And we want to move those contents toward the pyloric sphincter. Then we're going to move the fluid part of the chyme to be pushed toward the pyloric sphincter on the inside over here, whereas more of the solid center of the chyme is going to squeeze past the peristaltic contraction and move back toward the body of the stomach. Our peristaltic waves are going to move in the same direction and in the same way as the mixing waves, but they're going to be stronger. We have more of those muscles working in the musculars externa, contracting here and moving them down. And then we're going to have more of the fluid part of the chyme being pushed toward the pyloric region. You could see this at the blue arrows here, whereas more of the solid portion, once again, is squeezing past the peristaltic contraction back toward the body of the stomach. And then our peristaltic contractions are going to force just a few milliliters of the fluid chyme first through the pyloric sphincter, opening up, relaxing to move that into the duodenum. And most of the chyme, including the more solid portion over here, is going to be forced back toward the body of the stomach to mix it even more, and basically it'll become liquefied and continue into the duodenum as well. So next, let's discuss the small intestines. This is going to be the area of the greatest digestion and absorption of nutrients and water. So we mentioned in the beginning of our digestive system lecture that the small intestine is made up of 3 parts. We have the duodenum that is C-shaped over here, and this is the first 25 centimeters that can be found beyond the pyloric sphincter of the stomach. Then we see the jejunum here that makes up 2.5 meters of the small intestines, and the ileum will make up 3.5 meters and end joining the cecum of the large intestines at the ileocecal junction. We're also going to discuss how the ileum has an area called Peyer's patches or lymph nodules, and we'll see that in the coming slides. So the duodenum itself is going to curve over to the left, and we will see in another diagram that the head of the pancreas will be cradled by the duodenum. In this area too, which you will see an image in coming slides, we will have a major and minor duodenal papilla that will be opening of ducts in order to release contents from the liver, gallbladder, and pancreas into the duodenum. So if we look at the small intestines, we'll see that we have a large increase in surface area, about 600-fold, and that's possible by the following 3 structures here. The plicae circularis, or circular folds, so the tissue is folded up and that increases the surface area. If you look closer to these plicae circularis, we will see these finger-like projections sticking up, and these are villi. So this is one villi over here, and we have another villi over here. And this is containing inside of it capillaries as well as lymphatic capillaries known as lacteals. And then we have microvilli, which are going to be on the surface of the villi. They're going to be folds of the cell membranes of these absorptive cells. So let's look at the mucosa and the cells that make them up. We have absorptive cells in here. These are kind of the red cells over here. They are going to be the ones with the microvilli, and the microvilli will help to increase surface area and also help with absorption. The other job of the absorptive cells is to produce digestive enzymes. And then embedded in this mucosa, these darker spots in here are the goblet cells. And the goblet cells' function is to produce protective mucus so that we don't destroy the lining of the small intestines. And then we have endocrine cells. Usually you'll find most of those down in here, and they are going to secrete regulatory hormones such as the secretin and cholecystokinin that we'll discuss in a moment. And then we have granular cells, also known as Paneth cells, that will help us be protected from bacteria. Now we also have some intestinal glands, which you see pictured down here in this histology slide. And these are just tubular glands in the mucosa that we will find at the base of the villi. And below the mucosal layer, we will see the submucosal layer. And in here we have duodenal glands, or also known as Berners glands. And these are tubular mucus glands that are going to open up into the intestinal glands. So now let's discuss the jejunum and ileum. We'll see as we move from the duodenum through the jejunum and ileum that we will see decreases in the diameter of the thickness of intestinal wall. We'll also see a decrease in the number of the circular folds, the villi, as we move further from the stomach and toward the colon. This jejunum and ileum are going to be also major sites of nutrient absorption. And here we could see an area of the ileum that it has been transected. And you could see these Peyer's patches, which are patches of lymphatic nodules that are going to have lymphocytes within them to help with our immune system. And we'll find this embedded within the mucosa and the submucosa. Notice how our mucosa over here has those villi projecting in toward the lumen. Now, as we move past the small intestines, we are going to make it over to our ileocecal junction, basically where the ileum and the cecum are joining together. And I just noticed that ileum is spelled incorrectly here. It should be I-L-E-U-M. When we spell it with an I, that's referring to the bone ileum that is a part of the os coxa. So anyway, at this junction, we will have a circular muscle known as the ileocecal sphincter, so that we could control when the contents in the ileum move into the large intestines. And in there, we will also have an ileocecal valve to make sure that contents within the cecum do not go backward into the ileum. Instead, we continue moving the substances into the large intestines and through. So what are the secretions of our small intestines? We are going to have fluid in here that is made up of water, electrolytes, and mucus. We've already discussed how that mucus is going to help protect our GI tract, especially from the digestive enzymes and stomach acids. And then the digestive enzymes that we will have in the small intestines that are bound to the membranes of those absorptive cells will be your disaccharidases, the peptidases, and the nucleases. So our disaccharidases are going to break down disaccharides into monosaccharides. So that means from our 2 sugar molecules into 1 sugar molecule. And then our peptidases are going to help to hydrolyze the peptide bonds. So we are breaking peptide bonds between the amino acids. And then we have our nucleases that are breaking down our nucleic acids. We've already talked about our duodenal glands, but know that they are going to be stimulated by our vagus nerve and the hormone secretion and chemical or tactile irritation of the duodenal mucosa. So now we're going to get into our accessory organs of the digestive system. These are going to include the liver, gallbladder, and pancreas within the abdomen, and these are all associated with our small intestines. Know that we also have our salivary glands as accessory organs of the digestive system, but of course we're going to find that in the oral cavity. Now, let's start off with some main functions of each of these organs. So our liver's main function is going to be to produce bile, and we've already discussed that bile is going to be a fat emulsifier, so breaking those fat molecules apart from one another. And then our gallbladder's main function is going to be to store that bile. The liver is not going to keep it within itself, instead it moves it down here into the gallbladder. And then we have our pancreas, which is going to give most of the enzymes needed to digest the chyme in the duodenum, as well as bicarbonate in order to neutralize the stomach acid. So remember, as the chyme from the stomach moves into the duodenum, will be at a pH of less than 2. As the juices from the pancreas comes in, that will increase that pH to try to make it a little bit more alkaline. So more on our liver now. Let's look at the largest gland in the body. This is going to weigh about 3 pounds and has 4 lobes to it. So we have our right lobe here, which is the largest of the 4 lobes. We have our left lobe, found on the left side, of course. And then if we look at an inferior view of the liver now, so this is going to be my left side, this is my right side, this is anterior, and this is posterior. So we will first find the caudate lobe, which is located posteriorly. Caudate means tail, so the tail is found in the back, right? So this is at the posterior inferior portion of the liver. And then we have your quadrate lobe that is found anteriorly. And quadrate means square or four-sided. So you can see the 4 sides or square shape of this quadrate lobe. Now we have 2 ligaments to know on the liver. We have the falciform ligament that's going to be found between the right lobe and the left lobe, and this is extending out toward you because it's going to attach the liver to the anterior wall of the abdomen as well as the diaphragm that we would find superior to it. And then we have our round ligament. So the round ligament is going to be found here at the free edge of the falciform ligament, and this is remnant of our fetal umbilical vein. So this used to be your lifeline at one point. This is also known as ligamentum teres, and here's another view from this inferior aspect. So when we look at the liver, we will find a attachment to the stomach, and that is through the lesser omentum, and that isn't pictured here, but I believe I described it in part 1 of your lecture. So now let's talk about some vasculature here. We have the this area here where we see a bunch of vasculature and ducts moving in and out of the liver. So that is called the porta hepatis. And what that consists of is a hepatic artery, or sometimes we call this hepatic artery proper, and that is going to bring in oxygenated blood to the liver. Then we also have, I forgot I have some highlights here for you. Then we also have your cystic duct, and this is going to be the duct that is connecting to your gallbladder. And we also have in here your common hepatic duct. So that's going to be this portion right down in here, okay, past the cystic duct, and I'll show you a better image of the duct system below in just a moment. So here, I didn't highlight it, but this is going to be your right hepatic duct and your left hepatic duct. When those two come together, they create your common hepatic duct. And then we see the cystic duct coming off of the gallbladder. So when this common hepatic duct here joins the cystic duct, it becomes the common bile duct, which will then lead down to the duodenum to release the bile in there. So when we look at the liver tissue itself, we will see these liver lobules, and you could see that they have this wonderful hexagon structure to it. And we call these the functional units of the liver. They are composed of these hepatocyte plates. So you could kind of see this, the tan portion down in here. And the hepatocytes are liver cells, and their job is to filter and process nutrient-rich blood. And then the very center here, we have the central vein. So notice at the ends of the hexagon corners here, we have a lot of vasculature, and this is called your portal triad. So if we look at that, the ends of the portal, the ends of the hexagon, we are going to see the vasculature here, and that is called your portal triad. And the portal triad is made up of a branch of the hepatic artery that we see pictured over here. We said that supplied oxygenated blood. We'll also see a branch of the hepatic portal vein, which will bring in nutrient-rich blood. And then we will see the bile duct over here, which is going to receive bile from these bile canaliculi. And that will, of course, move on down the hepatic duct. So here's a little bit better view so you could see the interconnectedness of all of these, the neurovasculature and the ducts. So we also have these spaces over here that are venous, and we're going to call these liver sinusoids. These are leaky capillaries that are located between these hepatic plates, which are made up of the hepatocytes. And we will have blood from both the hepatic portal vein, which is right over here, moving up this way, and also blood from the hepatic artery proper percolate from the triad region, so you can kind of see this over here, through regions of the sinusoids to make it over and empty into the central vein. The central vein will then, through these interlobular veins, connect to the hepatic vein, which will take it over to your inferior vena cava. Now we'll also have in here your stellate macrophages or hepatic macrophages that are in the area of the liver sinusoids that are looking for debris or old red blood cells in order to get rid of them through phagocytosis. What do our hepatocytes do? They are going to produce around 900 milliliters of bile a day. They're also going to process our blood-borne nutrients. So for example, they will take our single sugar glucose molecules and link them all together to form glycogen to store that glucose for when we need it. And they can also make plasma proteins. They can store fat-soluble vitamins, so these would be vitamins K, A, D, and E. And they can perform detoxification, so converting ammonia to urea would be an example of that. Pictured here, we have a zoomed in look at a liver lobule. So in the center you see the central vein, and then the spaces that you see in here are the sinusoids, and in the pink we see sheets of the hepatocytes. So if I was to magnify this even more, these areas over here would be our hepatocytes. Now, what is bile really made up of? It's going to have a yellow-green look to it, and it is alkaline, which makes sense because as we move bile into the duodenum, we want to change the pH from in there from being really acidic to closer to the alkaline area on the pH scale. So within the bile, we have bile salts and bilirubin, cholesterol, triglycerides, phospholipids, and electrolytes. So first, what are bile salts? They are going to be derived from cholesterol, and they work to emulsify fat and to absorb. So if we take a fat globule here, these bile salts basically take a little piece of that fat globule and surround it in order to separate it from the other smaller pieces of fat, separating them out. Now, bilirubin is the pigment that is formed from heme, and we discussed it a little bit in our blood lecture already. So if you look at this figure over here, when we break down our red blood cells and we take this bilirubin from the heme that's derived from our hemoglobin molecule, we are going to move that into the large intestines, and bacteria in the large intestines are going to break it down to something called stercobilin, and this gives the feces its brown color. Now we can talk about an imbalance of the liver that will progress into a chronic inflammation called cirrhosis. And this could be from chronic hepatitis or from alcoholism. So what happens is the liver will turn fatty or fibrous, and also toward the end here we will have increased blood pressure or hypertension within those the portal vasculature. Liver transplants are going to be successful, but livers are hard to come by. But they can regenerate themselves to its full size in about 6 to 12 months after 80% of it has been removed. So pretty incredible. Now let's move to the gallbladder, and this is going to be a thin muscular sac on the ventral or anterior surface of the liver, and its job is to store and concentrate the bile by absorbing water and ions. We'll also take a look at the inside to see these honeycomb folds to it that'll allow the gallbladder to expand as we fill it with more bile. And then we mentioned that it has muscle within its wall, and that is used to contract the gallbladder to move the bile into its cystic duct, which will then flow into the common bile duct. So clinical application here is you may have heard of people complaining about gallstones or biliary caliculi. And this is caused by too much cholesterol or not having enough bile salts within their bile. And so this can obstruct the flow of the bile from the gallbladder into the cystic duct, and it is very painful when the gallbladder is trying to contract and move those gallstones into the cystic duct. So we can get something that develops called obstructive jaundice. So the blockage causes the bile salts within the gallbladder and pigments to build up within our blood. So everything is just backing up into circulation, and that'll result in jaundiced or yellow skin. Jaundice can also be caused by liver failure as well. And so the treatment for this, we have a couple different options, is we would take a drug in order to dissolve the crystals. We can use lithotripsy, which is sending ultrasound vibrations over to the stones and basically causing them to break apart into smaller pieces so they can more easily pass from the gallbladder into the cystic duct. We can also use laser vaporization or surgery. So now let's talk about the pancreas. Where we're going to find this is mostly retroperitoneal and deep to the greater curvature of the stomach. So you see the stomach over here, and really this is overlying on top of the pancreas. So we're going to see that the head of the pancreas is cradled by the duodenum, which is pictured over here, and the body of it kind of goes across the vertebrae that would be lying posterior here, and the tail, which is highlighted in teal here, is pointing to the spleen. Now the pancreas is really interesting because it both has a exocrine function and an endocrine function. And if you remember, whenever we hear the word exocrine, we want to think of ducts. And so if we look at the tissue here in the pancreas, we are going to have these acinar cells secrete enzymes. And these granules pictured here, these zymogen granules, are going to be the ones responsible for containing the proenzymes in order to make the enzymes. Then we will have these duct cells that will produce water and bicarbonate. And so by releasing the digestive enzymes with the water and bicarbonate, all of this will pass through the duct system over here, and we will kind of take a look at a grander scale of this in just a moment. And then the endocrine function it will have is to secrete insulin and glucagon in order to regulate our blood sugar, and that is secreted by our pancreatic islet cells. So these are also known as our alpha and beta cells of the pancreas. So here's our duct system. Let's just review real quick. Here's our right hepatic duct, left hepatic duct. Those join to create the common hepatic duct. Over here is our cystic duct, and when the cystic duct joins this common hepatic duct, it becomes the common bile duct, and that courses down and will release bile into the duodenum. So what we're seeing here, these small ducts will join together to the main pancreatic duct, and all of those digestive enzymes, water, and bicarbonate ions will move into the duodenum. So what is contained within the pancreatic juice that flows through that main pancreatic duct? Well, we said we'll have water in there and those bicarbonate ions, so that creates a alkaline solution of a pH of 8. and that will help to neutralize the acidic chyme that we would find in the duodenum. We'll also have some electrolytes in there, but the main electrolyte, of course, is our bicarbonate ion. Some other digestive enzymes that we'll find in there are proteases, and these are for proteins. We'll take a look at some specific ones in a moment, and they're going to be secreted in an inactive form. And the reason they are secreted that way is so that the pancreas doesn't dissolve itself. And then we have amylase, and this is for our carbohydrates. If you remember back to the oral cavity, we also have a salivary amylase. So this one we are going to call pancreatic amylase. And then we have lipases, and in this case we'll also call them pancreatic lipases because we also have some within our oral cavity that we call lingual lipases, and that is breaking down our lipids or fats. And then we have our nucleases that break down the nucleic acids within our food. Now notice we have these other enzymes here, trypsin, carboxypeptidase, and chymotrypsin. We're going to talk about this in just a moment, but this is assisting us in breaking down our proteins. They are grouped as proteases. And here they are. So we said they're secreted from the pancreas in an inactive form, and they're going to be activated once they reach the duodenum. So our enteropeptidase is an enzyme that is bound to the plasma membrane of the duodenal epithelial cells, and that is going to activate pancreatic protease trypsinogen. So here it is. There's our enteropeptidase coming from these epithelial cells, and we're taking trypsinogen, the inactive form, and changing it to trypsin. Now this is an important step because now trypsin can work to activate our other enzymes. So it's also going to go back and activate more of itself. So it goes back to trypsinogen, activates it to trypsin. It's also going to move down and take chymotrypsinogen and turn it into chymotrypsin, its active form, and go to our procarboxypeptidase in its inactive form and activate it to carboxypeptidase. So we discussed the duct system pretty thoroughly right now, but what happens once we get to this region over here? You will see that where our bile duct and our main pancreatic duct come together, they're going to fuse together in a bulb-like structure called the hepatopancreatic ampulla. And this ampulla is going to open up into our duodenum, right over here at the volcano-shaped major duodenal papilla. So that's just an opening into the duodenum. And then surrounding this area, really the ampulla portion, is going to be the hepatopancreatic sphincter. So you can see a little bit of the muscle that they've drawn in here around that ampulla, and that's controlling the bile and the pancreatic juice that's coming into the duodenum by contracting or relaxing. And you'll also see down over here we have the accessory pancreatic duct. This is just a smaller duct that is going to empty from the pancreas into the duodenum directly. So how do we regulate the bile and pancreatic secretions? They're both going to be controlled by neural and hormonal control. So the hormones that will help control this are cholecystokinin, or CCK, and secretin, and those are released by the duodenum. They'll move in through circulation and then go over to the pancreas to tell it to increase secretion, or we can inhibit the secretion as well when they're not present. So bile secretion is going to be increased when we have enterohepatic circulation, returning large amounts of bile salts. Also when we have secretin from our intestinal cells exposed to hydrochloric acid and fatty chyme over here, that'll go over through circulation to our gallbladder to tell it to release bile, as well as our liver to start creating more bile. And then we have our hepatopancreatic sphincter that is going to, I don't know if I necessarily want to say closed here, but rather that it is going to become relaxed so that we can move the contents in. Okay, so let's go through this diagram real quick because it summarizes those 3 points. So when we look at these epithelial cells in the duodenum, they are going to release the cholecystokinin or CCK based on receiving that low pH in here or the fats within the chyme. And our secretin will then be released from the acidic chyme, and they'll enter our circulation and move over to the pancreas first, and that will the CCK in here will cause the acid in our cells to create that enzyme-rich pancreatic juice to move into the main pancreatic duct, and secretin will cause secretion of the bicarbonate and the water to create that pancreatic juice. And then we also have the vagus nerve coming on down this way posteriorly in order to weakly stimulate the pancreas during that cephalic and gastric phase. So all that makes the pancreatic juice to be released in here. Then our liver is going to produce those bile salts returning from the enterohepatic circulation, which will be the most powerful stimulation for the bile secretion through these hepatic ducts, and we'll have secretin be a minor stimulus for that. Then as far as the gallbladder, we want this to contract because we already have bile stored in here. So CCK is going to cause that contraction, and we also see some fibers from the vagus nerve coming in over here and causing weak contraction of the gallbladder, or weak stimulus of the contraction of the gallbladder, and this is happening during the cephalic and the gastric phase. So those 2 components help move bile down into the duodenum here. And then we also have our hepatopancreatic sphincter that has to relax in order to get those contents into the duodenum, of course, and CCK is going to be the one to get that sphincter to relax. And let's move on to our large intestines now. So we talked about that ileocecal junction. We're going to start the large intestines from that point all the way to the anus. So it consists of the cecum, the colon itself, the rectum, and the anal canal. And movements in here are going to be pretty slow. So it takes about 18 to 24 hours. And at this point, the chyme from the small intestines is converted to feces. We're going to have absorption of water here and salts, secretion of mucus, and we have extensive action of our microorganisms. So a lot of gut microbes in here. We'll have about 1,500 milliliters of chyme enter into the cecum, but we'll see that 90% of the volume here will be reabsorbed back into our body, yielding about 80 to 150 50 milliliters left as feces. So let's talk about the different parts of the large intestines. Here is the cecum. The word cecum means blind, so I keep referring to this as being a blind pouch. And extending off of here, we will have the vermiform appendix. Vermiform means worm, so this is a worm-like structure. From the cecum, we're going to move up the right portion of the abdomen. So we're heading on up. If you're following with your hand, you want to start at the cecum at the right lower quadrant of your abdomen, and then move upward along that right side of your abdomen toward your next bend, which will be your hepatic flexure or the right colic flexure. This was called hepatic flexure because we see that it is below the liver on the right side of the abdomen. Then we're going to move across the abdomen over to the left side now in your left upper quadrant, and this is called your transverse colon. So in that the right upper quadrant, we will have the splenic or the left colic flexure, and this was named so because it's found right below the spleen. And then we will move down the left side of your abdomen down to the left lower quadrant, and this is your descending colon. And then this loop shape here that I think is kind of shaped like a backwards S is your sigmoid colon. And that leads to the rectum. Rectum means straight, so this is just moving straight down toward the anus. So we've already talked about your cecum and the vermiform appendix, but within your vermiform appendix, we are going to have a lot of lymph nodules, and so it plays an important role within our immune system because we will have lymphocytes within it. And then we discussed the different parts of the colon, but let's look at some of the tissue within the colon. We are going to have a circular muscle layer that is complete here. And we say this is really your muscularis externus layer. And we say this is complete because it goes completely around the large colon, whereas the longitudinal muscle in here is incomplete because we are going to only have these 3 Tenea coli bands. So you can see that kind of in this image over here. And those bands will also help to create these pouches that are called haustra. On the large intestines itself too, you'll see these fat pouches on here. These are called omental appendages, and right now their function is unknown. If we look in the lumen of the large intestines, the inner layer is called the mucosa, and as it dips down, it creates these crypts. And in the crypts, along with the mucosa, we will find these goblet cells, and we know that they are responsible for creating mucus. They are predominating over here, but we'll also have a lot of our absorptive cells as well as our granular cells, just as we did in the small intestines. So now we can move into the area of the rectum. We do have muscle surrounding the rectum, so we say it is a muscular tube, and that leads down to the anal canal. Now, if in the anal canal, the superior portion of this is simple columnar epithelium, whereas the inferior portion is made up of stratified squamous epithelium. And surrounding this area, we have 2 sphincters. We have the internal sphincter over here that's made up of smooth muscle, so that is involuntarily controlled. And then external to that, we have the external anal sphincter that is made up of skeletal muscle. So that obviously is going to be voluntary control, so you guys can decide when to relax and go to the bathroom. The other thing we have is if we have a lot of pressure or constipation in this area, that is going to put pressure on the veins, and that can cause the vein to enlarge or have inflammation. So if it's found within the anal canal, that is called an internal hemorrhoid. If it's found closer to the outside, that is called an external hemorrhoid. So we've already discussed the mucus within the large intestines, but know that it is parasympathetic stimulation that'll increase the rate of the goblet cells to cause that mucus secretion. We'll also have some pumps in that area. Bacteria will produce acid, and the following remove acid from the epithelial cells that will line the large intestines. So we can exchange the bicarbonate ion in there for chloride ions, and we can exchange the sodium ions in there for hydrogen ions. We'll also see that bacterial actions will produce gases from specific types of carbs that are found in legumes and also in artificial sugars like sorbitol. So I've pictured for you some foods that are high in sorbitol. And bacteria are also going to produce vitamin K, which is then absorbed. And our feces is made up of water, undigested foods such as cellulose from plants, microorganisms, or slothed off epithelial cells. Now, as far as the movements in the large intestines, they're going to be most common after meals, and they're integrated from our enteric plexus. We'll also have some local reflexes take place, which we'll discuss in the next slide. And this happens when we have food present in the stomach or the duodenum. So the reflexes are going to be called gastrocolic and duodenocolic reflexes, depending on where they are initiated from. And then we have the process of defecation. So our defecation reflex is when we have distension of the rectal wall by the feces moving into that area. This is controlled by parasympathetic stimulation and will be accompanied by voluntary movements in order to expel the feces. So to do this, we're going to create abdominal cavity pressure caused by inspiration and by contracting our abdominal wall muscles. So here's that image I was describing earlier. When we have food present in the stomach and the duodenum, we are going to have that gastrocolic reflex as well as the duodenocolic reflex stimulate the mass movements within the large intestines. And these reflexes are going to move the contents of the colon through toward the rectum. We'll also see once the feces are in the area of the rectum that we are going to have contraction of the colon and the rectum to move the feces toward the anus. We'll also have some local reflexes that will cause the internal anal sphincter to relax. We'll get our parasympathetic reflexes to go over to our spinal cord and move up our ascending tracts to basically communicate to your brain, hey, you really have to defecate, get yourself to a bathroom, and then we will get impulses or action potentials being sent down our descending tracts down the spinal cord and trigger our voluntary control to relax our sphincters and be able to move those feces out of the rectum and anal canal. So this section, last section, we're going to discuss on digestion and absorption. I'm just going to do a couple slides because you are responsible for reading through that section and then writing a paper on our digestive system assignment that I've posted under our Chapter 24 tab on Blackboard. So you guys will write a 1 to 2 page paper discussing food combining and what we just learned about our digestive system. So first I want to discuss the difference between the terms digestion and absorption. When we are digesting something, it means we are breaking down the food molecules in order to absorb it into circulation. So we have two forms of digestion. We have mechanical digestion, which we've discussed both of these before. Mechanical digestion is going to be breaking down the large food particles into smaller particles, and chemical digestion will be breaking the covalent bonds of those macromolecules by digestive enzymes. Now, absorption is going to mean that we take those broken down molecules and we're going to move it out of the digestive tract and into circulation so that we could distribute it to the rest of our body. So I really love this summary slide because it takes each of the macromolecules here and tells you exactly how it breaks it down. So we are going to see carbohydrates first digested within the oral cavity, and that's by use of the salivary amylase, and that breaks it down into polysaccharides and disaccharides. We don't really do anything to them within the stomach, but once we reach the duodenum, we are going to have secretion of that pancreatic amylase within the pancreatic juice that will break down those polysaccharides into disaccharides. And once we get into the rest of the small intestines, we will have those disaccharadases break it down from disaccharides into monosaccharides. So now when we look at the lipids, we are going to see it first digesting within the oral cavity through a lingual lipase. In the stomach, we have a gastric lipase. And then, of course, once we enter the duodenum, we will have the bile salts from the liver as well as the bile from the gallbladder move in, and our pancreatic lipase from the pancreatic juices in order to break down the lipids further into fatty acids and monoglycerides. Lastly, we have our proteins, and they don't get digested until we enter the stomach. In the stomach, we remember that we will have pepsin released, and that will break down the proteins into polysaccharides. Once we're in the duodenum, we will have those proteases activated into trypsin, chymotrypsin, and carboxypeptidases. And remember, those inactive forms were going to be released by the pancreas in its pancreatic juice in the duodenum. And that breaks down the polypeptides into peptides, and then the peptidases within the small intestines will further break it down into amino acids, dipeptides, and tripeptides. So that is it for your digestive system. Please let me know if you have any questions on our lecture or on your assignment.